So we'll start with Scott, who's here from Stanford, and he's done a great job on using and building and developing steady space models for integrating behavioral data and electrophysiological data. And I'm not going to hold you from hearing what he actually does.
All right, thanks so much. Sounds like this is working. Thank you so much for the invitation to be here. I'm really excited to tell you about some of our older and also more recent work on state space modeling in neuroscience. So the starting point for us is this very fundamental question of neuroscience. How does the brain represent and maintain internal states in order to produce appropriate behavioral outputs at the right time? And to make this very concrete and very much hypothetical, imagine that there's construction on Mercer Street. And traffic has backed up onto I-5, and you're on your way to work here at the Allen Institute. You're in your car. You've been patiently waiting in the exit lane to get off at your exit, and a car, some jerk, cuts right in front of you just before the exit. Now, maybe that morning you had a nice night's sleep, you had a nice cup of coffee, you're feeling pretty good, and you think, wow, that guy, he must be really late for work. He's probably really stressed, and you let it go. And if that's you, I applaud you. I aspire to that level of zen in my life. Maybe, however, you're a little bit more stressed out. You didn't get such a nice night of sleep. You already spilled your coffee on yourself once, and this guy cuts you off, and you respond with a auditory output via your horn and maybe a physical output with one of your fingers. That's another type of behavioral output that you might manifest depending on what internal state you happen to be in. And I think for us as neuroscientists, a really exciting question is how is the brain representing and maintaining such states? And we have a lot of tools now with which we can try to answer those questions. Tools, many of which have been developed and pioneered and used here at the Allen Institute for recording very large numbers of neurons simultaneously, methods for tracking behavioral outputs with unprecedented resolution, and very excitingly, tools for actually going and causally testing predictions about how systems work using closed-loop patterned optogenetic stimulation, like we'll see a bit today. So I want to tell you about one example of how my lab, in collaboration with others, is trying to answer these types of questions. And it's particular, the question we're interested in is how the brain represents and maintains an internal state of aggression in mice. And I'll tell you about some of the methodological work that we've done to try to answer this question using recurrent switching linear dynamical systems. I know some of you, I recognize many people, you've probably heard me talk about these before, but I want to describe how these ideas work and for those who maybe aren't familiar. And then I want to tell you about some exciting experimental results using these methods in closed loop to test hypotheses about circuit dynamics, and time permitting at the end, tell you about how we've closed the broader scientific loop, having used these methods to test different hypotheses. That's shown us many ways in which these methods could be improved, and we're taking steps to do exactly that. So before I go any further, I want to make sure that when I say we in an experimental context, I really mean they, in particular, Adi, who is a grad student, now postdoc with me in David Anderson's lab. And this work started when Adi was a grad student with David and working with Ann Kennedy, who's now at the Scripps Institute. So the setup comes from work from David's lab that's gone on for over a decade now, where they've been interested in the neural basis of social behaviors, and in particular, aggressive behaviors between male mice. And so in this setting, this is a video of experiment done long ago where they optogenetically stimulated a region of the brain called the ventrolateral subdivision of the ventromedial hypothalamus. And what you can see here is that when that light turned on, this mouse went from being totally calm, cool, collected, getting along fine with its neighbor, to all of a sudden going ballistic. And this led to a very clear hypothesis that this particular subpopulation of neurons in this small subregion of the hypothalamus is essentially an on-off code for aggression. You stimulate these, you get aggressive outputs. Now, fast forward a few years, and when they took a closer look using miniscope imaging in this same population of cells, they found a more nuanced picture. In fact, what they found was that there's a broad tuning of activity within this subpopulation. Some neurons that are indeed very much active when the mouse is attacking, but other neurons that are not really tuned to attack, maybe, if anything, more tuned toward passive, more sniffing-like behaviors, and then a whole lot of cells that are somewhere in between on the spectrum. And so this led to a somewhat more refined hypothesis, the idea that this state of aggressiveness is encoded in this region, but it's done so via the collective activity of these cells, not just simply the overall activity of the whole population. And so the question that we sought out to answer is how could we test this using some of these probabilistic modeling tools we've been developing? In particular, how do we formalize this hypothesis? So the idea is as follows. Take that miniscope recording, it's neurons by time. We're going to think of this, this data matrix, and in particular a column of this data matrix, as encoding the neural population state. It's a length-end vector that says how active is each neuron at this point in time. And as time unfolds, you can think of this neural state as tracing out a trajectory in a very high-dimensional, n-dimensional space. And so if this population is encoding something quite low-dimensional, like a graded signal of aggressiveness, then that would suggest that the collective activity doesn't span all of n-dimensional space. Rather, it lies much closer to a low-dimensional manifold, something you might think of conceptually like this gray plane that's only two-dimensional and embedded in this much bigger n-dimensional space. And so the way we think about this neural trace is really like a noisy readout of a lower dimensional trajectory, this green trajectory that lies on this plane. And the points on this plane that are closest to that neural trajectory are what we call the continuous latent states, the coordinates on this manifold, if you will. Now, what's more, we're interested not just in inferring these latent trajectories, but also the flow fields that govern how they evolve over time. And these flow fields you could think of or visualize as a little vector field that tells you if you're at this point at time t, where do we think you're going to go at time t plus 1? And why we're interested in those flow fields is because various hypotheses about how neural circuits compute have been articulated in the language of these flow fields. So rotational dynamics have been postulated to underlie the various forms of motor control, providing rotational or oscillatory bases for making a variety of movements. Point attractors, like you see in the top right, have been hypothesized as mechanisms for encoding memories. Saddle points, like you see in the bottom left, could be important for decision making. And line attractors, which are going to be the star of today's talk, have been postulated as a method for performing some form of integration, keeping track of a one-dimensional variable that can move up or down along this line. And the methodological question is, how do we go about trying to infer these latent states? How do we estimate those dynamics functions, those flow fields, from high-dimensional neural time series, like what I was just showing you? And the way that we do so is using what are called probabilistic models. And if you've not seen a picture like this on the right-hand side, this is a graphical model. The nodes in this graph are random variables, and the ones that are filled in are things we observe, the neural activity, for example. The ones that are unfilled are the things that we want to infer, the latent states, the dynamics function, et cetera. And the arrows tell us about the relationships between these variables. What do you need to know in order to generate the next latent state, x at time t plus 1? Well, you need to know where you are in state space, and you need to know what that dynamics function is. So that's how you read this graph. And the question at this point is, well, what types of assumptions, what types of models should I plug in for that dynamics function f? And here I'd argue that there's really a spectrum of choices you could make. You could choose a very simple, say, linear model for that dynamics function on the one hand, or on the right-hand side of the spectrum, maybe something that's much more flexible and expressive, like a neural network. And these come with various trade-offs about capacity, but also efficiency that we're going to be balancing as we move along this line. So what can linear models on the left side of this spectrum do? Well, it turns out they can do a whole lot. So all of these motifs that I showed you here are all instances of linear dynamical systems. F of x is ax plus b. And with models like these, we argue we can interpret these dynamical systems. We can find things like their fixed points in analytical form. We can get stationary distribution, dynamics along eigenmodes. We can think about how to optimally control these systems to drive them into a desired state. So there's a lot we can do with linear models. But of course, there's a lot of things that you can't do with linear models, and by far, most computations require some form of nonlinear dynamics. So if you want bistability, then you need to have some nonlinear dynamical system, 2 point attractors separated in space. If you want a ring attractor, you need some form of nonlinear dynamical system. Now, important thing to note here is that while these systems are nonlinear, oftentimes the way we analyze and think about nonlinear systems is by trying to break them down into a composition of different linear pieces. So this bistable system may be well approximated as linear on one or the other side of this space. The ring attractor is maybe linear inside and linear outside, and the composition together gives you this nonlinear dynamical system. And this is exactly the idea behind a class of probabilistic state space models called switching linear dynamical systems. And going back to that graphical representation, the way these look is pretty much what we had before, but with the addition of this extra set of discrete latent states, z, evolving up top. And the role of these discrete states Z is to tell you which of your linear dynamical systems, red or blue, for example, to use at each point in time. And these models have a very long and rich history in a variety of disciplines spanning decades now. We need some rule for how those discrete states are updated. The simplest assumption is to assume that they follow a simple transition matrix, like what you see on the right. But one of the things that you'll immediately notice is that that is an insufficient assumption about how these discrete states evolve. And in particular, for this picture like what you see here, in order to capture this nonlinear dynamical system, you need to know two things. You need to know, where you are in this continuous state space in order to know which of your linear dynamical systems to apply. And that wasn't present in the simple SLDS. And so a line of work that I've been interested in working on for many years now is what we call recurrent switching linear dynamical systems that address this problem with a very simple assumption. We just add an extra set of arrows, these red arrows going up and to the right. And the way to think about this is it allows you to partition state space up into regions with different linear dynamical systems, because now you know where you are in state space, you can figure out which system to apply. So going back to this spectrum, the way that we think about these is essentially moving us to the right on this spectrum, giving us a little bit more flexibility, but not sacrificing too much in terms of interpretability and ease of fitting. So Adi, the graduate student in David Anderson's lab, used these RSLDS tools to analyze those miniscope recordings from before, and what he did, he found something pretty interesting. So first of all, a large fraction of the variance in these traces can be explained in terms of just a handful, say two-dimensional latent variables. And then in this two-dimensional latent state space, the trajectories follow a pretty interesting dynamical system. Upon the entrance of this intruder mouse, there's a big blip that kicks it off into the top, and then it comes quickly down into this trough, where it moves slowly but steadily to the left of this flow field. And the way you can think about this flow field, another way to think about it is in terms of an energy landscape, where you imagine a ball rolling around in this energy landscape. It falls down into this trough and then slowly moves along the bottom of this trough. And as it does so, the outward overt behaviors of this mouse become increasingly aggressive. It starts out sniffing on the one end, but over time, this translates into more dominance mounting, attack, et cetera. And so we can decode, based on position on this line attractor, what types of behaviors this mouse is likely to be exhibiting. Now, what's really nice about the RSLDS analysis is that it's broken down that landscape into regions with different linear dynamics. And in particular, that trough is well explained by one of these linear dynamical components. And as I said, with linear dynamical systems, we know how to analyze their behavior. Importantly, we can go in and take that dynamics matrix, look at its eigenspectrum, and what we find is there's one eigenvalue that has a much longer time constant. It corresponds to a much longer time constant than any of the others. And this is the hallmark of line attractor dynamics. And indeed, when we project activity onto that one dimension, what we see is a nice ramping up of neural activity spanning hundreds of seconds, and it's correlated with this increase in aggressiveness of behavior. And importantly, when the intruder mouse is taken out, the state doesn't just decay back to zero. It persists for tens of seconds up to minutes after the exit of that, which is kind of consistent with how we feel after somebody rudely cuts us off. You probably don't just go back to your Zen-like state. You're probably still a little frustrated for a while, hopefully not too long. The other thing I want to mention is that this is not true of all regions of the brain. It's not even true of all hypothalamic nuclei. If you look next door in MPOA, you see very different dynamical systems. Now, that work still leaves open an important question. We're suggesting that this region of the hypothalamus is encoding this type of aggressive internal state. But is that where it's actually maintained, or is this just a readout of some upstream region that happens to be doing the interesting work? And so this is where emit comes in. We wanted to be able to do the following dream experiment. You'd like to be able to instantiate this line attractor-like dynamical system in a head-fixed mouse where you can optogenetically stimulate neurons and test the predictions of this model. Can I move the state of this system along the line? If I kick it off the line, does it decay back down? Now, unfortunately, when you head-fix a mouse, it's not very inclined to attack other mice. So in order to get this type of dynamical system, we needed some other approach. And the breakthrough happened actually with a result from Nirao Shah's lab at Stanford in 2023, where what they found was this sort of mirror neuron property. The same neurons in this region that are activated during these aggressive interactions are also active when the mouse is viewing other mice fighting one another. And so this allows you to set up a system where the mouse can be head fixed, but watching other mice involved in this attack and still get the same types of dynamics out. And so that was the first thing that Amit did. He tested this, recording with mini scopes and freely moving mice, seeing these line attractor dynamics, and then looking at the same mouse in the head fixed viewing setting and found that you could see the same types of attractor dynamics, largely overlapping sets of neurons that are performing this or that are involved in this dynamical system in both settings. And so now we could actually do the test. Go in and stimulate subsets of neurons in order to try to move this system along the line or off the line. Now, the hypothesis here is that if we pick out a handful of neurons, 5 cells that we think are most aligned with the line attractor dimension, and we stimulate those, well, if this line attractor type dynamical system is really instantiated in this population of neurons, then when we stimulate those, you should move along that attractor, that trough, and you should stay wherever you end up. However, if it's just a readout, then when you stimulate, you're going to get kicked back down, and so the stimulation shouldn't have a long-lasting effect. It should look more like what you see on the right. And indeed, when we do this stimulation, we pick out the 5 cells that are most projected along this line. We see a nice integration effect. There's a bit of a blip and decay back down, but the net effect is after many sequences of stimulation, we can move this state into this elevated position along this line, i.e. we get this integration-like effect. And I want to point out that this is echoing one of the things Jakob said. This really required us to fit this model to a very small amount of data. In a single session, we needed to fit the model to like 10 minutes of data, make a prediction based on the model fit of which neurons would get this or would drive the state in this way, and then design this holographic perturbation in order to test that model. The control, of course, is if we pick 5 neurons that are actually projecting on the other axis of the state space, we should see transients, but they should decay back down to the line very quickly. Indeed, we see very quick decays in this off-manifold direction, as we call it. And if you prefer to look at this in the landscape of this dynamical system, this is what it looks like. So on the on-manifold, moving it along the lines, these pulses move it over to the right. There's a small decay back down, but by and large, the system moves to the right and stays there. And then if we get to the end and we stimulate those other 5 neurons, we can move it off the manifold, but it quickly decays back down. So these types of perturbations that are enabled by this closed-loop model-based prediction of which neurons to stimulate allow us to get, I think, really exciting causal evidence of these dynamics with really fine-grained, only a handful of neurons being stimulated. Now, I think I have 2.5 minutes left, so I want to spend just a minute telling you about how this line of work has led to more methodological work within the lab, because this was all based on these RSLDS models that we've been working on for a long time, but of course, within this collaboration, there have been plenty of places where like, I wish we could have done that a little bit better. And so one example of that comes from this work with graduate student Amber in my lab and postdoc David, former postdoc Leah, who's now at Columbia. And the idea is that we'd like to be able to get some uncertainty about that dynamical system, right? This flow field, we've treated it so far as just a thing that we estimated and was fixed. But of course, that's not the case. We have a small amount of data. There's lots of different flow fields that could produce the same types of trajectories. And so we'd like to be able to quantify how certain or uncertain we are about those dynamics at each point in state space. And so the idea is to model that dynamics function f using a Gaussian process. And this Gaussian process is going to have the advantage that it's going to be able to capture these same types of piecewise, like switching linear dynamical systems. We constructed the kernel to be able to do exactly that. So we get the same type of flow field, but we also now can put this gray shading on it to say, here's where you should trust the flow field and here's where you shouldn't. And what's more, we can quantify under the posterior distribution of flow fields, given the data we've observed, here's how probable we think it is that each point in this state space is an attractor, is a slow point. So this is work that came out just last year, and now we have lots of ongoing collaborations with David's group to put these to use in some of the follow-on works for that line attractor work. One of the other things I want to emphasize in the context of this workshop in particular is that this is all made possible by our software engineering efforts and our open source engineering efforts to build great tools for fitting dynamical systems models. One example of this is Dynamax, which just was published in JOS last week. And this is a project that started when I was doing a visiting researcher position at Google and working with Kevin Murphy and a team of Summer of Code students to try to write JAX code. Again, plugging some of the things that Jakob mentioned before. JAX code that allows us to really execute and fit these models very efficiently on whatever hardware you have available. And really to make that connection even stronger, so we've actually been working with Jakob and one of his students, Michael, who developed Jaxly, to use Dynamax to do state inference of the voltages and channel states over time in biophysical models given very noisy voltage recordings. So I think it's a really great synergy that we're able to go back to back here. So just to conclude, understanding how the brain performs state-dependent computations, I think, is a really fundamental question for neuroscience and one that we have a lot of great tools to start to tackle. State space models, I think, are an important path toward answering those questions, pulling out these low-dimensional structures in high-dimensional data, and then closing the loop and asking, based on our model fits, how would you design a perturbation, optogenetically or other, in order to drive the system and see if the predictions of your model are borne out? And a lot of this hinges on algorithmic work and modeling work that allows us to fit these things fast in real time in order to do this type of closed loop science. So I want to thank all the members of my lab, especially I mentioned Amber and David and Leah, and none of this would obviously be possible without amazing collaborators, David, Adi, Amit, and Anne. So thank you, and happy to take any questions.
David.
Hi, I had some technical questions. So the first time you showed data from Adi, you showed plain two principal components. Okay, but and the claimed attractor was moving just along one dimension. But was that at the border between two of these different linearized regions?
Yeah, maybe I can go back. So the model fit, let me see if I can do this quickly.
I mean, the first thing you did was a piecewise linear analysis.
Yeah, so there's two
where's
dimensions.
The border?
So yeah, the borders are not shown, and I guess this is not going to do anything to help you see it probably. There is probably a line here and a line here. The borders themselves are, I think, something that we aim to quantify that uncertainty in the GPS LDS work, because largely, I think you could fit this model and get one linear dynamical regime that really captures most of the attractor, but the entrance to the attractor is maybe just slightly oriented a bit in a curved way that it splits into two pieces.
So, I mean, I guess the question is, it seems that this attractor is running along the border. Is that correct?
It's not running along the borders, no. It's definitely within. It's just there's a split at one point in between. And you can also see that in this space here where, I guess on this one, here we end up with like a border, but this GPS LDS model, I didn't give you enough detail to say that there's not like a hard border in these systems. It's by design constructed in such a way that it kind of smoothly interpolates between the linear dynamics of one system and another.
The other thing, the other technical question was, at one point you showed the eigenspectrum of the different decays. One was like 100 second.
Yeah.
And the others were longer. And then you showed data where you removed the aggressor. And things seem to stay constant for hundreds of seconds, which is much longer than this decay mode. So,
well, it's not longer than the decay mode. It's kind of on the order of the longest mode. So the longest time constant there was on the order of 100 seconds, and that's about what we see when the intruder is taken out. I think the important point there is that these time constants are much longer than the time constants of individual neurons in this population, and certainly longer than the time constants of the calcium indicators that are being used. And so we think that this really has to arise from some sort of circuit level mechanism that's able to persist this state. And here we have some evidence that that's intrinsic to VMHVL, but in other work that I think is also quite relevant and is something we're also exploring more, there are other components of this. There's hormonal drives, there's neuropeptidergic inputs, there's lots of stuff that collectively can produce these long time constants.
All right, Eric, and then Carl, and move on. Christopher.
Absolutely beautiful. I'd like to ask you about two possibilities for describing these types of low-dimensional dynamics. One is to have a state space which is explicitly low-dimensional, as in your work or the work of Tatiana Engel or others, which is marvelous and successful here. The other, maybe more like Jacob Macca, would be to have some sort of low rank or other special parameter structure, but to have the dynamical model actually exist in the full dimensional space in which the recordings or maybe even the brain is unfolding. Can you tell us if probabilistic inference methods like you're developing have a shot at the second approach as well, and your comment on the relationship between the two overall?
I think it's a great question. I also think it is very closely connected to these questions of how we go about perturbing these systems, because when you really design these perturbations, you're acting on cells, right? You're acting on neurons, and these models, as fit so far, I made an inductive leap that wasn't necessarily justified from the generative model, that neurons that are correlated with this one dimension are, if we stimulate those, likely to move us along that dimension. So I think one of the directions of future work that, certainly something we're exploring, but I think I'd be remiss if I didn't mention like Sergej Stoyak's work toward these ends as well. Your work too, I think, is asking, how do circuits based on their recurrent neural networks, based on their properties of their weight matrix, produce these types of low-dimensional phenomena?
All right, Carl.
I have a maybe slightly related question. So this happens over tens to hundreds of seconds, and of course the inputs to the system change, and you're kind of modeling this with a constant landscape. But clearly the landscape is not constant, right? So, and when I look at your perturbation experiments, as a beautiful experiment, very difficult experiment, it really looks, glass half empty, half full, it more looks like a moving fixed point rather than a line attractor, which is, of course, something, as you know, in ramping in motor cortex, that's how we interpret what things go. You perturb, things snap back, you still have ramping, right? But so how do you think about it? Clearly that landscape is not constant over hundreds of milliseconds. You mentioned peptides and changing inputs and all this kind of stuff.
So I think there's a really interesting question there, and I guess I can say a couple things. So one is we are looking over much longer time scales. So one of the experiments is, hunger and aggression often go hand in hand. So, as your hunger state evolves over hours, you would expect this landscape to also change. And so for that, as we're recording throughout, up to 40 hours of food deprivation, you can see the landscape itself viewed through these RSLDS lenses changing with that hunger level, the hanger state, as we call it. But you're also right, like the neuropeptidergic inputs, what's the right time scale to think about that? Is that something that's changing over hundreds of seconds? And should we treat these as changing the nature of the dynamical system, like there's a flow field that's a function of the neuropeptidergic input, or should we think about it as a fixed flow field, but with an extra, input in the form of like slowly varying neuropeptidergic signals that are driving it? I don't know if there's what the right answer is there yet, but I do agree that there's room for improving that aspect of this model, because you're absolutely right. We're treating it as fixed when it's almost surely varying over some time scales. It's just a question of like which time scales are most important. Thank you.
Let's thank Scott.